Hi everyone, it's Mr. Sinti, and this lesson is on the cytoskeleton. And as the name sort of implies, cytoskeleton is a skeleton within the cell. So cyto meaning cell, skeleton. But it's not a skeleton like our own skeleton for our body, which is constructive of bone. It's actually a skeleton inside the cell, but it's constructed with threads of protein. And it's rather important. And you're like, well, what's going on with all this protein? It sort of looks real busy in here in the cell. There's all sort of thick filaments and thin filaments and intermediate filaments. And for one thing, right out of the gate, the function of the cytoskeleton is very much like our own skeleton, which is to provide structure for the cell. Without these protein sort of girders inside or beams, the cell would be rather mushy, and as you know that we're not that mushy. And if you start stacking cells on top of each other, they would sort of collapse without this protein radiating outward, sort of providing stability for the cell. But what I want to talk about is some of the really interesting things in addition to them providing stability and support for the cell. These proteins can elongate and contract. In other words, they can get they can go out a little further and then come back. And what's interesting is when they attach to the cell membrane, they can do different things like pull the cell membrane in and push the cell membrane out. So you might be thinking, why would you even want to do that? And so let's take a look over here. This is a video that I want to show you. This is a actual blood under the microscope. These sort of circular looking cells or red blood cells, they carry oxygen. They don't move so much even though the blood is moving because it's under the pressure of the heart beating. But this particular cell right here, this really large guy, is a white blood cell. And this is not sped up or it's not time-lapse photography. This is real footage, a digital camera on top of the microscope filming this. So this is a white blood cell. This is bacteria. I want you to check this out. And so when I click this, notice how the white blood cell is moving. It's sort of crawling along, or if you will, creeping along, and it's like chasing after the bacteria. It's going along and it's able to move itself. And it's like, well, how is it able to move itself? Like, well, our body is capable of moving itself because our muscle cells are made up of these proteins inside. There's a tremendous amount of cytoskeleton in our muscle cells. As you may know, those protein fibers can contract and relax, and so that ultimately shortens the muscle and elongates the muscle, and because it's connected to a tendon to a bone, we're able to move. But likewise, this cell, a white blood cell, is very mobile because inside it has these proteins inside. And by pushing on the cell membrane, it's actually able to sort of scoot along and travel. Assured, it's not a great distance, but it doesn't need to go that far. But it's chasing after bacteria. And then when it actually catches up to it, it's going to stretch out its membrane through phagocytosis or endocytosis and take it in. And then you may remember this from a previous discussion. That bacteria is then in what's called a food vacuole. And then that goes to a lysosome where it's digested. And so white blood cells, because of the fact they're moving along, literally save our lives every day because bacteria are trying to infect us. And so these white blood cells, with their cytoskeleton, are able to sort of push and push and push and push. Because the cell membrane itself is a phospholipid bilayer. And so really where it's pushing is on proteins. And so it's protein pushing against protein. So I just wanted to point that out. I think that's kind of cool. So let's go back to the slideshow here. And let's go here, if we can. Enlarge it. So let's go to the next slide. So the cytoskeleton. So this is, if you think about it, this is obviously a cartoon drawing of a cell, but you just saw a real cell moving around. And the reason that the real cell was moving around is that it has all of these protein fibers in there. And so here's the cell membrane, and the cytoskeleton provides support and it maintains the shape of the cell. And it also is an anchor for other organelles. And so it's sort of like the mitochondria, the endoplasmic reticulum are sort of held in place by all these threads. And what's interesting about it is some of the fibers, as you can just simply see, are really thick. The thickest ones are these things called microtubes or microtubules. There's intermediate ones, which obviously are intermediate in size, and then there's these thin filaments called microfilaments. And so these things can push or tug on the cell membrane, which allows the cell to sort of move in and out. And it's like, well, why else would you want the cell to move around? Say you were a cell and you were really big and you needed to divide. And so it's like, well, I have a feeling the cell divides by having its membrane pinch in. And then it'll eventually divide into two cells. And you're like, well, how does that happen? And it's like, well, that happens because these thin filaments, these microfilaments in here, I'm just going to highlight them in green, they connect to the cell membrane like this. And so they're sort of like a purse string. When you pull on these proteins right here in this direction, you can actually get the cell membrane to pinch in. Now this is three-dimensional. So it's kind of like a water balloon, if you will, that's getting pulled in. And so ultimately, if you pinch it tight completely, it'll break into two cells. And so that's really important in cell division. And so there's a lot of movement involved in cell division. And so what's interesting, this little foreshadow to a future conversation, is cell division is sometimes called cytokinesis. And I like that because kinesis, as you may know, means movement. And so cyto, movement. So the cell dividing is all about cell movement, and that's accomplished by microfilaments pinching in. So that's pretty cool. Now, what's interesting here is before the cell divides, let me sort of highlight this. But here's the cell membrane. And you're like, okay. And what's interesting here is what you didn't see before this picture is, let me draw the nucleus. If I were to draw the nucleus like this, and the DNA is located here and here, and you know, notice how there's two groupings of DNA. But once upon a time, there was only one DNA. But before a cell can divide, it needs to replicate its DNA so that there's twice the amount, because if there's going to be two cells, obviously you're going to need two copies of DNA. So this is what has happened before this picture. There was a nucleus, there was DNA replicated itself, and then what happened? Check this out. The nuclear membrane then dissolves, and because you're not going to be able to move the DNA with the nuclear membrane. And so you got these two pieces of DNA, and you're like, well, I wish one of them would go to the other side of the cell, because if the cell's going to divide, one of the DNA wants to be on the left side, one wants to be on the right side. If they're both on the left side, or both on the right side, or both on the top, both on the bottom, this is a problem. So it's like, okay, DNA, get over to the other side. It doesn't just go by itself. It needs to be pulled. You need some sort of protein fibers. Oh, cytoskeleton might be involved. You need to pull the DNA. Sort of like if you wanted to like catch a fish, you needed a fishing line to pull and reel the fish in. And so check this out. Let me erase this DNA over here. And so now what you're actually seeing here is this is the DNA, and this is the DNA in a cell that's getting ready to divide. Notice it's not dividing just yet. It's still one cell. And it's like, well, how are we going to get the DNA to move around like that? Check this out. There's a type of protein called microtubules. Remember, these are the thickest ones. You can literally see them in this picture here under the microscope. These threads, like this, are pulling the DNA in this direction and pulling the DNA in this direction, And so when you pull the DNA apart from one another, that allows for the DNA to separate on opposite sides of the cell. So check this out. Then the cell membrane, whoops, I didn't want to do that. The cell membrane then wants to start to pinch in, and it's safe to do so because the DNA has sort of been partitioned. Now, if these are microtubules, let me just go with red here. The microfilaments, or the thinnest ones, are the ones that are pulling here to make the cell divide. So it's really cool. The cytoskeleton are these proteins inside that help move DNA. They help to move the cell. They help the cell to pinch in. And I might just add something here. Let me sort of click off of this and show you. I have a little animation of what I was trying to describe. Hopefully it'll work. So this is a process, you may recall it somewhere in your past. This is what I was trying to describe, where the DNA is being pulled apart. This happens to be called anaphase, which is one of the phases of mitosis. Check it out. And so here's the DNA, and it's all wrapped up in a chromosome so that it's easy to travel. And see these threads in here? These are the microtubules, and they're literally pulling the DNA apart. And let me just pause it for a second. The reason they're pulling the DNA apart like this is because each individual cell, here's the whole cell, each cell doesn't need 2 copies of DNA, it only needs 1 copy of DNA. So these threads, these microtubules, you may have remembered them called spindle fibers. They're often called spindle fibers, and in some circles they're called polar fibers because they emanate from or originate from the poles. You might be wondering what's going on right here. These little structures are called centrioles, and they're made up of microtubules, and they're the ones that can radiate or contract. In other words, polymerize or depolymerize these microtubules. And by shortening, they pull, and by elongating, they push. So that's kind of clever. And so the cytoskeleton is involved in movement of DNA inside the cell because the cell is about to divide. So I hope you enjoyed that. So let's go back over here. Let's see if I can manipulate this properly. Let's go here, and then we'll go to our, let's go, here. Yes, okay, thanks. So here's another picture, different colors, but here the DNA is in blue, and the microtubules are shown here in fluorescent green, which is kind of neat. So the microtubules, which are green, are made up of a bit of a detail, but in some instances it's not a detail, but I'm just throwing it out, that all these proteins have names. And so this particular microtubule is made up of a protein called tubulin. And tubulin, like any other protein, you can make more tubulin and add them together, sort of like beads on a string, and the microtubule will elongate, or you can remove the beads and they can shorten. So that's important, and they help to move chromosomes during mitosis. And so that's kind of cool. I was mentioning these structures on the ends of the cells where the microtubules are radiating. These two structures are called centrioles, and each animal cell has a pair of them, so that's 2. And they're usually in one side of the cell, but that's not going to be that effective, because if you think about it, when the cell is about to divide, you don't want these centrioles to be just on one side. You want some to be on this side and some to be on the other side so they can pull the DNA apart. Think about that. And so what happens during the process of mitosis is that these centrioles duplicate, so there's a total of 4 of them. And this is a term that is not as important, but centrosome is the area of which they are. And what happens is, if I can animate it, the protein just simply elongates from the centriole. This is an actual transmission electron micrograph of centrioles, so we know what they really do look like this. And so the centrioles come out like this, they radiate their spindle fibers or microtubules, and then they grab the DNA in the center. That's kind of cool. Let's go over here. And so here's another thing. I mentioned this before about cilia, which are these proteins on the outside of the cell. And this is transmission electron, this is scanning electron over here. And these help cells move also. And so though I was saying that the cytoskeleton is protein within the cell, sometimes it can be extended to the outside of the cell, that these proteins on the outside of the cell are involved in cell movement. And so cells can have cilia, which help them to move around. So proteins are all about movement. So the cell moving, DNA moving, and even within the cell, if I can go back a slide, even within the cell, let me animate this if you don't mind. Here's a cell right here. These are kind of like bricks. There's a cell here. Even if there's a transport protein that needs to move around inside the cell, sometimes the transport proteins are moved along these cables. So they're like little monorails where molecules can move inside the cell. And so that's kind of a cool thing. I have a video of that coming up in a moment. So here's some cells moving around. This is cilia, this is a flagella, and here's male reproductive cells, which need to be mobile in order to reach the female egg for fertilization to take place. And so let me pause it here, go here, go here, and I can show you some flagella instead of just talking about them. Over here, this is a single-celled protist that's capable of photosynthesizing. I know that because it has green chloroplast. What's interesting is you can see its flagella moving around. And so, granted, it's not really moving quickly, but it's capable of some motion because its flagella is sort of contracting, relaxing, sort of like our muscles. And so it's able to propel the euglena around ever so elegantly, I would say. So let's go back here. Let me sort of take you to this conversation. Sorry about that. I'm just trying to figure my way through here. Let's talk a little bit about, if I come up here and then I go down here. Sorry. Let's talk about plant cell walls. Plant cell walls are rigid. They're on the outer side of the plasma membrane. Prokaryotes like bacteria have them. Fungi have them, which are eukaryotic cells. Even some protists, but they're mainly found in plants. Well, I can't say mainly because the most numerous organisms are prokaryotes, but we commonly think of a cell wall as being part of a plant cell. So it's on the outside, and you're like, what does it do? It provides support. And so the fact that wood is very strong and we can sit on it or we can build houses out of it is because wood is made up of plant cells that are very strong cell walls. And like, what makes it so strong? It's made up of this polysaccharide, as you may remember, called cellulose. And cellulose is chains and chains of glucose. And so what's the difference between cellulose and starch? It's this sort of beta orientation or this up orientation. And so it's very difficult to break down. Like, for example, fiber is not something that water can easily break down. If we wanted to, if a tree was made up of just starch and it started to rain, the starch would obviously sort of digest and hydrolyze and like soggy bread. The plant would sort of just collapse. And so you need something a little stronger than starch. And so cellulose provides a polysaccharide that's rather rigid, and so that's important. And then lastly, and I'll show you some videos to summarize, the outside of the cell is referred to the space outside the cell is called the matrix, and so it's called extracellular, like outside of the cell matrix. And so, like, what's on the outside of the cell? Well, this is the outside of the cell. Here's the cell membrane, so this is inside. So you got these microfilaments, as I mentioned before, which are connected to the membrane, so cell can pinch in. But then look at this. On the outside of the cell, you've got carbohydrates, which are important in identification, and you've got these big columns of protein on the outside, and that helps for cells to stick together and make the tissue very strong. You may have heard of this protein called collagen. It's found in our skin and many other places in the body. Like, for example, it's found in tendons and ligaments, and it helps that particular tissue to be very, very strong. You need a tendon to be strong, and you need ligaments which connect bones to be very strong. So what gives those cells extra strength? It's not the cell itself, It's the protein between the cells. And so that's called the extracellular matrix. And so check this out. Let me come down here and show you what I'm talking about over here. So here's a little video of the extracellular matrix. I find this to be really well done. So check this out. This is one cell membrane and another cell membrane. And so let me pause this here. What's happening is all these threads are collagen fibers out here. These purple things are proteins embedded in the cell membrane. So these proteins help connect cells to other cells, so making a formidable barrier so cells don't move around. And then notice here this green guy looks like some kind of hormone that's binding to a protein. We haven't talked too much about that. Hopefully this will play if I can click the play button. What's interesting is you could see that some of these proteins act as channels where ions go through. And so let's actually travel inside that channel there. We'll go inside, ooh, cytoskeleton. So notice here the thick filaments and the thin filaments. And there's some mitochondria. Let this play out. Push play. And so you can see how transport vesicles are being moved along in some of those corridors there. And that's what I wanted to show that. And then finally, we have here this video to sort of conclude on the cell wall. Here obviously is we're going inside of a leaf, and if we were carbon dioxide, this is how we would enter into the underside of the leaf through those pores called stomata. And then let me pause it right there. I'm going to go back just for a second. So these are individual plant cells. So that if we were going to bump into a plant cell, the first thing that we'd bump into is the cell wall. So check this out. So the cell wall, and you're like, wow, I didn't realize the cell wall looked like this. These are columns and columns and columns, not of protein. It's kind of funny. We were talking about the cytoskeleton, which, if you will, looks like this, but this is not protein. These are the threads of cellulose that make up the cell wall. And so as we peer through the cell wall, then we would go through the cell membrane, and then we'd actually enter into the plant cell. And you might recall they have this big swimming pool called the central vacuole and these green chloroplasts. Plants also have a cytoskeleton. So you can see transport vesicles moving along there. And so I hope you enjoyed our conversation on the cytoskeleton, and thanks for watching.